Hello, our final lecture on Alzheimer's disease is going to talk about animal models and biomarkers and therapeutic approaches to Alzheimer's disease. Experimenters in modern neuroscience have a series of animal models for Alzheimer's disease, especially mice that overproduce APP, amyloid precursor protein, and other mice that have mutations in presenilins, again, especially with Alzheimer's disease associated mutations. So there is some progress, and the challenge is to make data from mice relevant to those with people. Now, in general, what does one want to do for Alzheimer's disease? Well, as for Parkinson's disease, which we'll discuss in the next series, you would like to do two things for Alzheimer's. You would like to find a person with early stage Alzheimer's and stop that early stage Alzheimer's from progressing any further. And if you get it early enough, why, that's a remarkable change in the time course of the disease if you can stop it from progressing. The way to stop it from progressing early enough is either to have a very good neurological or psychiatric diagnosis, or to have a so-called biomarker, that is, an objective measurement, either biochemical or physiological, that tells you, yes, this person has very early stage Alzheimer's without a doubt. Now, we are not far along on either of those challenges. Some of the new data suggest that if you take measurements of the tau, remember the tau protein, and phosphorylated tau, and of A-beta 42, and look at the individual values or the ratios among these, you can get a fairly early diagnosis of Alzheimer's disease. Another fruitful approach has just been approved by the FDA, the U.S. Food and Drug Administration. It's a molecule called fluor-beta-PIR. It seems to bind to amyloid plaques that contain beta-APP. Here is its structure, and here is the F18, which emits beta particles. That's why it's called fluor-beta-PIR. So here is a typical positron emission tomography. A person who does not have Alzheimer's disease, very few plaques, very few places, black places, for fluor-beta-PIR to bind. Here is a person who, by other criteria, does have Alzheimer's disease. Clearly, there's a lot more binding of fluor-beta-PIR. So one needs to be extremely careful about these scans, as they're called. And here are the words that the manufacturer uses. A negative scan, sparse to no neuritic plaques, inconsistent with the diagnosis of Alzheimer's disease at the time the image is made. So probably that patient does not have Alzheimer's disease. Since a patient goes to a neurologist in the first place for cognitive impairment, among other complaints, then that cognitive impairment is not Alzheimer's. Now, of course, one needs to be very careful and say that a positive scan indicates moderate to frequent amyloid plaques, and that some patients with these kinds of plaques also have Alzheimer's. But there are examples of people with other types of neurological conditions, as well as older people with normal cognition who show positive scans. So the manufacturer says very carefully, fluor-beta-PIR is an adjunct to other diagnostic evaluations. More than this, we can't say at the moment. We hope for better biomarkers. Again, heterozygote advantage does not seem to exist for Alzheimer's, so we'll leave that go until we get to depression. And let's look now at therapeutic approaches. Suppose a person is diagnosed with early stage Alzheimer's, what can be done? Well, the molecules that are in the clinic now, and that are the molecules of choice, are the acetylcholinesterase inhibitors, because neurons that make acetylcholine are among the first to die in Alzheimer's disease. They're in the basal forebrain, and therefore if one can prolong the life of acetylcholine by inhibiting its breakdown by acetylcholinesterase, one has more acetylcholine, and the relative lack of cholinergic neurons does not matter much. The three that are on the market are somewhat effective. Interestingly enough, galantamine may also be a positive allosteric modulator of alpha-4 beta-2 nicotinic receptors, so that it could be acting both by preventing the breakdown of acetylcholine and by enhancing its action. Also on the market is one NMDA inhibitor called memantine, and the thinking here is that excessive activity at NMDA receptors may be excitotoxic, and so it may be possible to slow down the time course of Alzheimer's using memantine. For both of these types of medications, the slowing down is statistically significant, but we need very much better drugs. So, because we've talked about the various secretase sites in beta-APP, you can appreciate that beta secretase inhibitors would also be useful if one could develop them with no side effects and specific action. There have been other drugs with unknown mechanisms that failed for Alzheimer's trials. Gamma secretase inhibitors might also be useful. The one that got the furthest along in clinical trials also failed. One could think about targeting the protease complex with notch-sparing gamma secretase inhibitors. Remember, the probable natural function of gamma secretase is to proteolyze notch, so you'd need to develop a gamma secretase inhibitor which allows notch proteolysis to go ahead, but blocks cleavage of APP. And other gamma secretase drugs, modulators that shift the proportion of A-beta peptides in favor of shorter, less harmful peptides, have been conceived but not brought to the clinic. And then there's the topic of antibodies against a beta-APP or A-beta. And the three that have been tried so far in clinical trials are listed here. They have been largely unsuccessful, although one of them has been mildly successful in early stages of Alzheimer's. So clearly Alzheimer's therapy has a very long way to go. Next time, we will continue our discussion of neurodegenerative diseases by talking about Parkinson's disease.